Today we will be starting off with synapses. Can you tell me what is a synapse?
A synapse is the gap in between an electrical impulse is converted into a chemical form.
Okay. It's a gap. It's a synaptic cleft. When two neurons meet, they do not quite touch with each other. There is a very small gap, about 20 nanometer wide, and between that gap, this gap is called as the synaptic cleft. The parts of the two neurons near to the cleft plus the cleft itself, ये एक जो रीजन है ये वाला, इसको हम कहते हैं साइनैप्स। अब ये पहला न्यूरॉन है और ये दूसरा न्यूरॉन है। ये पहले न्यूरॉन की माइलिन है, एक्सॉन है, उसमें माइटोकॉन्ड्रिया भी है और प्लस कुछ प्री साइनेप्टिक वेसिकल्स भी हैं। जो यहाँ से न्यूरोट्रांसमीटर वाला सिग्नल आ रहा है उसको इन द फॉर्म ऑफ वेसिकल्स उसने यहाँ से यहाँ भेजना है। इस मेम्ब्रेन को हम कहते हैं प्री साइनेप्टिक मेम्ब्रेन और दूसरे न्यूरॉन की मेम्ब्रेन को हम कहते हैं पोस्ट साइनेप्टिक मेम्ब्रेन। फर्स्ट न्यूरॉन इज नोन एज द प्री साइनेप्टिक न्यूरॉन, बीच वाले गैप को हम कहते हैं साइनेप्टिक क्लेफ्ट और एंड पे फिर आपके पास आता है पोस्ट साइनेप्टिक न्यूरॉन। Now, what's the mechanism of this synaptic transmission? Now, we know कि impulses cannot jump across the type of synapse. Axons के ऊपर तो समझ आती है, they can jump और वो saltatory conduction के through the whole mechanism. बट एक पूरी इम्पल्स जो इन द फॉर्म न्यूरो ट्रांसमीटर्स आ रही है, ठीक है, वो हमें चाहिए होंगे। वी हैव टू कन्वर्ट द इम्पल्स इनटू न्यूरो ट्रांसमीटर जो कि एक न्यूरॉन से दूसरे न्यूरॉन तक ट्रांसफर हो जाए। सो व्हाट इज अ साइनेप्टिक क्लेफ्ट? It is a very small gap between the two neurons at a synapse where the nerve impulses are transmitted across the synaptic clefts by neurotransmitters. A synapse is a point at which two neurons meet, but they do not touch. The synapse is made up of the end of the presynaptic neuron and the synaptic cleft and the end of the postsynaptic neuron. Neurotransmitter is a chemical released at the synapse जो कि impulses transmit कर देता है from one neuron to the other neuron. So here is an outline of the sequence of the events that actually occur और ये किस तरह से ट्रांसफर होता है across the synapse. सबसे पहले an action potential occurs at the cell surface membrane of the presynaptic neuron. अब मैं यहाँ पे न्यूरॉन बना देता हूँ, कोशिश पूरी है। ये आपके पास है प्रीसाइनेप्टिक न्यूरॉन एंड देन यू हैव द पोस्ट साइनेप्टिक न्यूरॉन। ठीक है, दिस इज प्री साइनेप्टिक, दिस इज पोस्ट साइनेप्टिक। हुआ ये है कि एक्शन पोटेंशियल यहाँ पहुंचा है प्री साइनेप्टिक न्यूरॉन के ऊपर। नाउ द एक्शन पोटेंशियल कॉजेज द रिलीज ऑफ द मॉलिक्यूल्स ऑफ द ट्रांसमिटर सब्सटेंस इनटू द क्लेफ्ट। ये एक्शन पोटेंशियल ने क्या किया है, इन वेसिकल्स को, उस इम्पल्स को इन द फॉर्म ऑफ वेसिकल्स यहाँ पे फ्यूज करवाया है ताकि वो इस साइनेप्टिक क्लेफ्ट के अंदर आ जाए। Now the molecules of the transmitter substances diffuse across the cleft and they go and bind to the receptor molecules जो कि present है आपके पास post synaptic membrane of the neuron के ऊपर। पोस्ट साइनेप्टिक न्यूरॉन के ऊपर जब ये आके बाइंड कर जाते हैं उनके रिसेप्टर्स पे, इसको हम कहते हैं कि डीपोलराइजेशन हो चुकी है और अब यहाँ पे एक्शन पोटेंशियल जनरेट होगा और आगे इम्पल्सेस सेंड हो जाएंगे। Let's now look at these processes in more detail. Now, the cytoplasm of the presynaptic neuron contains the vesicles of transmitter substance. More than 40 different transmitter substances are there. For example, noradrenaline and acetylcholine are found throughout the nervous system, whereas others such as dopamine, glutamic acid, gamma-aminobutyric acid, GABA, ये कुछ brain में होते हैं. You don't have to remember the names except acetylcholine or noradrenaline. You will concentrate on the synapses that use ACH as the transmitter substance. We call it the cholinergic synapses. अब ACH को हम एज अ एसिटाइलिन कोलिन को हम एज अ ट्रांसमिटर स्टडी करेंगे जो हमने उस दिन नोट से भी किया था. So you will remember that as an action potential occurs at one place on an axon, the local circuits depolarize the next one piece of the membrane as well, which stimulate the opening of the sodium ion channel proteins. Now, in the part of the membrane of the presynaptic neuron that is next to the synaptic cleft, arrival जैसे ही होगा एक्शन पोटेंशियल का। अब आपने ध्यान से सुनना, ठीक है? दिस इज वेयर द डिटेल स्टार्ट्स। दिस इज द प्रीसाइनेप्टिक न्यूरॉन एंड दिस इज द पोस्ट साइनेप्टिक न्यूरॉन। अब ये तो था आपका बेसिक जो हमने पढ़ा था, द बेसिक इवेंट सिग्नलिंग। अब हमने पढ़नी है इसी की डिटेल। एक्शन पोटेंशियल आ गया, ठीक है? एक्शन पोटेंशियल हैज अराइव्ड एट द प्रीसाइनेप्टिक न्यूरॉन, जिसने क्या किया है, causes the voltage gated calcium ion channel proteins to open. यहाँ पे इस प्रीसाइनेप्टिक न्यूरॉन के अंदर क्या खुल गए, कैल्शियम चैनल्स। इसकी वजह से क्या हुआ है, द कैल्शियम आयंस डिफ्यूज इनटू द साइटोप्लाज्म। सोडियम आयंस तो आ ही रहे थे, वो तो हर एक्शन पोटेंशियल के अंदर आ जाते हैं, लेकिन यहाँ पे एक एक्स्ट्रा काम ये हुआ कि कैल्शियम आयन चैनल प्रोटीन्स भी खुल गए हैं, और सिर्फ सोडियम आयंस अंदर नहीं आए, but also the calcium ions diffuse into the cytoplasm of the presynaptic neuron, and there are virtually no calcium ions in the cytoplasm but many in the tissue fluid. सारे जो कैल्शियम आयंस थे वो यहाँ पे सराउंडिंग में थे, तो जैसे ही चैनल खुला वो सारे कहाँ पे आ गए, presynaptic neuron के अंदर, but many in the tissue fluid surrounding the synapse. This means that there is a very steep electrochemical gradient for calcium ions, इसलिए वो बड़ी आसानी से high to a low gradient अंदर आ गए presynaptic neuron के. Now the influx of the calcium ions stimulates the vesicles containing ACH. अब जो न्यूरोट्रांसमीटर का नाम है वो आपने याद रखना, दैट इज व्हाट, एसिटाइल कोलिन. जैसे ही कैल्शियम आयन अंदर आते हैं ये उन वेसिकल्स को, और उन वेसिकल्स के अंदर कौन सा न्यूरोट्रांसमीटर है, एसिटाइल क्लोरीन, जो कि हमारा न्यूरोट्रांसमीटर लेके जा रहा है, जो कि हमारी इम्पल्स लेके जा रहा है. They empty their contents into the synaptic cleft and the attachment of the vesicle to the membrane is held by the snare proteins. कुछ snare proteins होती हैं जो हेल्प करती हैं ACH को प्री साइनेप्टिक से निकलने के लिए. Each action potential causes just a few vesicles to do this, and each vesicle contains up to 10,000 molecules of acetylcholine, and ACH diffuses across the synaptic clefts in less than 0.5 millisecond. अब जब वो क्लेफ्ट के अंदर आ गया, अब उसने आगे कहाँ जाना है, पोस्ट साइनेप्टिक न्यूरॉन के ऊपर, और वहां पे कुछ लगे हुए हैं आपके लिए रिसेप्टर्स. The cell surface membrane of the postsynaptic neuron contains the receptor proteins. Part of the receptor protein molecule has a complementary shape to the part of the ACH. So what is the presynaptic neuron? A neuron ending at a synapse from which the neurotransmitter is released. Postsynaptic neuron जहाँ पे action potential arrives. Noradrenaline and acetylcholine are called types of neurotransmitters. Cholinergic synapse जो भी हम पढ़ रहे हैं वो कौन सा है, जहाँ पे न्यूरोट्रांसमीटर ACH है. कहीं पे किसी पार्ट में जैसे एड्रिनल ग्लैंड्स हो गए वहां पे नॉर एड्रिनल होता है, तो उसका एक डिफरेंट रिस्पॉन्स का नाम होता है. सो अब हुआ क्या है, अब जब वो वेसिकल्स रिसेप्टर्स पे आए हैं, ACH molecules can temporarily bind with the receptors, and the binding changes the shape of the protein, opening the channels through which the sodium ions can pass. अब सोडियम आयंस जो होते हैं वो तो देखो हर जगह होते ही हैं, जहां एक्शन पोटेंशियल होता है वहां सोडियम आयंस तो होते ही होते हैं, तो वो भी डिफ्यूज कर जाते हैं अंदर की तरफ, कहाँ पे, पोस्ट साइनेप्टिक न्यूरॉन में डाउन द इलेक्ट्रोकेमिकल ग्रेडियंट, और अब पोस्ट साइनेप्टिक को वो डीपोलराइज कर देते हैं. These receptor proteins with their channels are ligand-gated ionic channel. They are stimulated to open by, not by a voltage change. If ACH remain bound to the postsynaptic receptors, these sodium ion channels will remain open. जितनी देर तक ये ACH लगे हुए हैं ना रिसेप्टर्स के ऊपर, सोडियम आयंस अंदर आते रहेंगे. ACH is also recycled to prevent this from happening and also to avoid wasting it. क्योंकि ACH तो एक ट्रांसपोर्ट है ना, जिसमें हमारी इम्पल्स बैठ के साइनेप्स क्रॉस कर रही है. The synaptic cleft contains an enzyme, acetylcholinesterase, which catalyzes the hydrolysis of each ACH molecule into acetate and choline as well, ताकि हम इसको स्टोर कर लें, और जब हमें दोबारा ACH चाहिए होगा तो हम इन दोनों को मिला के ACH एक दफा से फिर बना सकते हैं. So this enzyme in the synaptic cleft on the postsynaptic membrane hydrolyze कर देते हैं ACH को acetate and chlorine में स्टोर कर लेते हैं अगली दफा के लिए. This choline is taken back into the presynaptic neuron where it reacts with acetyl coenzyme A, और दोबारा से ACH को बना देते हैं और फिर से एक दफा रेडी हो जाते हैं for the initial arrival of the next action potential. Got it?
Yeah.
अब एक दफा समझते हैं. एक्शन पोटेंशियल आया, जब एक्शन पोटेंशियल आता है एक चीज तो कंफर्म होती है कि आपके पास सोडियम आयंस तो हर जगह ही आते हैं, जहां एक्शन पोटेंशियल होगा वो अंदर आ गए डाउन द इलेक्ट्रोकेमिकल ग्रेडियंट. साथ ही साथ एक्शन पोटेंशियल की वजह से क्या हुआ, कैल्शियम आयन चैनल भी खुल गए, और वो भी अंदर आ गए कैसे, हाई टू लो इलेक्ट्रोकेमिकल ग्रेडियंट. अब हुआ ये कि आहिस्ता आहिस्ता अंदर वाली साइड अब आपका डिफरेंस इन एक चार्जेस आ गया, एंड व्हाट हैज रिजल्टेड कि जो ये एक्शन पोटेंशियल आ रहा था वो इन द फॉर्म ऑफ ACH न्यूरोट्रांसमीटर इन वेसिकल्स के अंदर जमा हो गया. ठीक है, क्योंकि कैल्शियम आयंस अंदर आ गए थे, अब वो वेसिकल्स सारे आ गए. प्लस यहाँ पे इस प्री साइनेप्टिक न्यूरॉन में एक प्रोटीन है स्नेयर प्रोटीन. स्नेयर प्रोटीन इन न्यूरोट्रांसमीटर्स की मदद करती है यहाँ मेम्ब्रेन के साथ फ्यूज होने में, कि बाहर निकलो जल्दी से. और वो बाहर निकल आते हैं। जब वो बाहर निकल आते हैं तो यहाँ पे वो रिसेप्टर्स के साथ बाइंड होना शुरू करते हैं। एक रिसेप्टर, दूसरा रिसेप्टर, तीसरा रिसेप्टर, चौथा रिसेप्टर, पांचवा रिसेप्टर्स। और जैसे जैसे न्यूरोट्रांसमीटर्स रिसेप्टर्स के साथ बाइंड करेंगे, आपके पास सोडियम आयंस अंदर आते रहेंगे। साथ 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 ही साथ क्या क्या हो रहा है, सोडियम आयंस भी अंदर आते जा रहे हैं। और आपको पता है कि जब सोडियम आयंस अंदर आ गए तो आपके पास यहाँ पे भी एक एक्शन पोटेंशियल जनरेट हो गया। अब जो ACH इनको लाया था, उस ACH को हम एंजाइम के थ्रू वापस एसीटेट और कोलिन के अंदर कन्वर्ट कर दे, ताकि हम आइंदा इसको दोबारा से कोएंजाइम A के साथ रिएक्ट करवा के दोबारा यूज कर सकें एज अ ट्रांसपोर्ट। गॉट इट?
Yeah.
So an action potential arrives. Depolarization of the synaptic terminal stimulates the opening of voltage-gated calcium ion channel proteins and the calcium ions diffusion. Synaptic vesicles move towards the presynaptic membrane. ठीक है, ये presynaptic membrane है, ये postsynaptic membrane है. Synaptic vesicles fuse with the presynaptic membrane and empty the ACH into the synaptic cleft. ACH diffuses across the synaptic cleft and the molecules bind to the receptors in the postsynaptic membrane. Then the receptors go through the conformational change and open to allow sodium ions to diffuse into the postsynaptic neuron. और ये भी आपके पास depolarize हो सकता है। जब फिर हमारे पास एक एंजाइम होता है जो कि ACH को ब्रेक डाउन कर देता है इनटू एसीटेट एंड कोलिन, और कोलिन को दोबारा प्री साइनेप्टिक न्यूरॉन एब्जॉर्ब कर लेता है, ताकि वो उसको कोएंजाइम ए के साथ मिला के एक दफा फिर से ACH बना सके फॉर द नेक्स्ट इम्पल्स जो कि अराइव करेगी एट द प्री साइनेप्टिक न्यूरॉन। इज इट ऑल क्लियर? Is it all clear?
Yeah.
Good. Now, the depolarization of the postsynaptic neuron leads to the generation of an action potential when the potential difference is above the threshold for that neuron. If not, then there is no action potential. यहाँ पे भी वही बात है, एक proper threshold reach करना जरूरी है. And the chance that an action potential is generated and an impulse is sent to the postsynaptic neuron is increased if more than one presynaptic neuron releases ACH at the same time over a short period of time. So much of the research on synapses has been done at synapses between a motor neuron and a muscle, not those between two neurons. Motor neuron forms a motor end plate with each muscle fiber, and the उस synapse का नाम होता है neuromuscular junction, जो कि mostly muscles और motor neurons के अंदर पाया जाता है. Such synapses function in the same way as described above, and an action potential is produced in the muscle fiber, which may cause it to contract. So what is a neuromuscular junction? ये जो ज्यादातर हमने पढ़े ये वो मोटर न्यूरॉन्स है जो मसल्स में पाए जाते हैं. तो हम जो साइनैप्स हमने पढ़ा है सिलेबस के अंदर वो कौन सा है? न्यूरोमस्कुलर जंक्शन, which is a synapse between a motor neuron and a muscle. और सबसे ज्यादा रिसर्च भी इसी साइनैप्स के ऊपर की गई है. So can you tell me what do you think is the role of synapses?
Synapses, like, they're responsible for transmission. And other than that, do one-way transmission to you, that they're responsible for that. And they also connect nerve pathways.
Okay, good.
And we're back. The team is looking down the barrel of a 4:55 meeting. Hi guys, this video will be
An introduction to synapses, the presynaptic bulb, the synaptic cleft, the postsynaptic neuron, and then we'll finish with a summary. So a synapse is an important structure in the nervous system. When you see a synapse, it's where we link two or more neurons together. So obviously the body is made up of lots of different neurons and lots of types of neurons, and because they're carrying communication, they need to be able to bridge to each other. And the way that they bridge this is at a synapse. And you can think of it as a junction between one neuron and the second neuron. And it can also link more than two neurons together, so it can have several neurons receiving information from the same neuron. So just to illustrate here, we would have a gap between the end of one cell and the start of the next cell, and this would be what this structure looks like. The purpose of the synapse is to allow the action potential which is propagating along to travel from one neuron to the next, and it's communicated across this gap. So it basically allows the information that's coming along to travel over the gap and to the next neuron. The information comes along and it travels from the presynaptic neuron across the synaptic cleft to the postsynaptic neuron. So there's a few specific terms you need to know here. We've got three elements across the synapse. First of all, we have the synapse before the neuron, so we call it the presynaptic neuron. It then has to cross the gap between the two neurons, and this gap is called the synaptic cleft. Cleft meaning a kind of gap between two things. And then finally we have the last element, which is the postsynaptic neuron, the cell after the synapse, hence the post before its name. And that would be this neuron. So whenever you write about synapse in an exam, it's really important to be specific in using these terms so that the direction of the information travelling is clearly represented. So first of all, let's talk about the first element of the synapse, which is called the presynaptic bulb or presynaptic knob. It's basically a swelling at the end of the axon, and it's the axon of the presynaptic neuron. So remember, we've got action potentials travelling down this axon and arriving at this swelling, which is the bulb, and this is the presynaptic neuron. So the synapse is slightly thicker than the axon itself, and it tends to be filled with some synaptic vesicles, and they're membrane-bound vesicles filled with neurotransmitters. So here's the synaptic bulb, and here you can see these spherical vesicles, and they're basically membrane-bound, and they're containing particular chemicals. So the membrane obviously surrounds the cell as a normal cell membrane would, but you can have internal membranes as well, like vesicles, carrying these chemicals. And in this case, at the synapse, the chemical that these vesicles contain are called neurotransmitters, which are important for the function of the synapse. So the function of neurotransmitters, for example, one of these types of neurotransmitter is called acetylcholine. They're basically particular chemicals that stimulate the postsynaptic neuron. So the presynaptic neuron has these vesicles, and across the synapse, the vesicles are going to release this neurotransmitter to go into the cleft. And the neurotransmitters are going to go across this synaptic cleft and act at the postsynaptic neuron, and then stimulate it so that it can then carry its own information onwards. So they're the kind of bridge between the presynaptic neuron and the postsynaptic neuron. The vesicles are membrane-bound, and they're made in the smooth endoplasmic reticulum. So remember, the smooth endoplasmic reticulum is an organelle. The smooth endoplasmic reticulum, or SER, is an organelle found in the cells, which has multiple functions, and one of its functions is to make membrane-bound vesicles. So these vesicles are made at the SER, and therefore they go down to this synaptic bulb to be ready to sending out these neurotransmitters. When these vesicles are being released from the presynaptic neuron, they require ATP to do this. So the synaptic bulb not only contains the endoplasmic reticulum, but it also contains mitochondria in the presynaptic bulb. So here we can see a mitochondrion, which main focus of the mitochondrion is to produce ATP, which then drives the release of the vesicles and the spread of neurotransmitters out to the synaptic cleft. And the final feature of these synapses is that the synaptic bulb has voltage-gated calcium channels present in its membrane. So looking again at the presynaptic bulb in the cell membrane towards the end, we see these proteins embedded in the membrane, and these are voltage-gated calcium channels. So remember not to get mixed up between different voltage channels. These are carrying calcium ions through them, not sodium or potassium. And the purpose of these proteins in the membrane is to detect when an action potential arrives at the end of the axon. Because when an action potential arrives at the end of the axon, it's signaling that this message needs to get across the synapse, and then that detection that this has arrived will go through these channels. So the next part of the synapse which we need to discuss is the synaptic cleft, which is the gap between the pre and the postsynaptic neurons. The neurotransmitters get released from those vesicles from the presynaptic bulb, and then they go into the synaptic cleft. So up to this point, what we've had is an arriving action potential triggering the release of these vesicles, and as the vesicles get released into the membrane, the neurotransmitter gets released into the synaptic cleft, the space between the two neurons. The synaptic cleft is very, very narrow. It's about 20 to 30 nanometers wide, and this is about the width of a bacterial flagellum. So remember, bacteria are much smaller than eukaryotic cells, and the width of that flagellum is about the same as the width of this synaptic cleft, about 20 to 30 nanometers. And this is useful because the minimum amount of space means this travelling distance is minimized, and therefore the information can quickly get from one cell to the other. As well as there being spreading neurotransmitters in the synaptic cleft, also contained are enzymes which help to break down the neurotransmitter. Because when that neurotransmitter is finished with, the enzymes present in the cleft need to break it down so that it doesn't keep firing across. So here we have enzymes ready to break that neurotransmitter down and stop the signal. And finally, the postsynaptic neuron is the last element we need to discuss. Once the neurotransmitter has diffused across the synaptic cleft, it reaches the cell membrane of the second cell, which is the postsynaptic neuron. So as you can see, as the neurotransmitter goes across the cleft, it reaches the second cell, which is only 20 to 30 nanometers away, and this is the postsynaptic neuron. And what we find on the membrane of the postsynaptic neuron facing this direction are proteins called neurotransmitter receptors. So remember, in a cell membrane, we can have certain proteins embedded in, and sometimes these proteins are receptors. And they're proteins designed to respond to particular chemicals that can bind to them, and then this sets off some sort of response. So in this case, these channels here are neurotransmitter receptors, and so they're going to respond to the presence of these neurotransmitters in the cleft once they arrive. The receptors themselves aren't just receptors, they're actually specialized ion channels that close or open when a neurotransmitter binds to them. So there are different types based on the different neurotransmitters, but it can either be open or it can be closed. And the point is, it allows the transport of particular ions in and out of the cell depending on what one it is. In one example, we can have what's called a cholinergic synapse, where the neurotransmitter is acetylcholine, and on the postsynaptic cell we have an acetylcholine receptor opening if the acetylcholine binds to it. So let's say that we have the postsynaptic cell membrane here, and inside the cell membrane we have this neurotransmitter receptor, in this case the acetylcholine receptor. And when it's not bound to acetylcholine, the channel is closed. However, if the presynaptic neuron has released vesicles containing acetylcholine, the acetylcholine is going to diffuse across the cleft and then bind to the receptor, and in doing so, it's going to open the channel. Now, this ion channel can allow the movement of particular ions in or out of the postsynaptic cell, and therefore set up the correct response for the information to travel or not. In the case of the acetylcholine channel, this causes sodium to enter into the cell. So once this channel has been activated, after acetylcholine has bound, sodium ions from the cleft diffuse into the postsynaptic cell. And this will trigger the setup of a new action potential in this cell, and so the information can travel onwards. Hey guys, I hope you enjoyed the video. If you were
All right, are synapses clear now? Pakka?
Yes, sir.
Good. So, talking about the role of synapses firstly slow down the rate of transmission of nerve impulse that has to travel along two or more neurons, और responses to a stimulus would be much quicker अगर directly transfer हो जाते. Number one, synapses also ensure one-way transmission, that they can only pass in one direction at synapses. This is because neurotransmitter is released on one side and its receptors are on the other. There is no way that the chemical transmission can occur in the opposite direction. Moreover, synapses also allow the interconnection of nerve pathways and allow a wider range of behavior that could be generated in a nervous system in which neurons were directly wired up to each other. They do this by allowing the interconnection of many nerve pathways, और वो किस तरह से होता है and what's the whole smooth muscles, arteries or pressure wala. We'll start that in the very next lecture where we talk about the different neurons, dendrites, and how they provide a large surface area in the muscles and in the brain. और a synapse के ऊपर you have to write a whole paragraph, और मुझे आपने भेजना है of this whole process that you can see in this particular diagram, क्योंकि ये 4 marks का सवाल आता है और एक ही है, और इसके अलावा इस टॉपिक का कोई सवाल नहीं है. So you have to write a whole paragraph for these questions and then send me by tonight. Got it?
Question number 9A and 9B.
Kind of, yeah.
Okay.
हाँ बल्कि वही हो गया. Yeah, that's all.